Good morning, all. Good morning, everyone. Good morning. Great, great, great. Good morning. Okay, so friends, in the yesterday class, I mean to say, in previous class of nitrogen metabolism, we were discussing about the nitrogen cycle, and friends, we understand very clearly the various steps involved in the nitrogen fixation, and we have finished even the first step of the nitrogen fixation also. If every one of you quickly recall about the nitrogen fixation, we read that the most important enzyme that is involved in this particular nitrogen fixation is the nitrogenase enzyme. And friends, we also read very clearly that if the nitrogenase enzyme wants to be active, always the bacteria has to maintain the anaerobic conditions. We also read, friends, how the bacteria are going to maintain the anaerobic conditions. Later, we started studying about the establishing the symbiotic relationship. So, while studying the symbiotic relationship, we understand the types of genes involved. So we read clearly, friends, there are different type of genes are involved in the root nodule formation. I hope all of you remember. We read majorly nodulin genes, nod genes. We can also call it as what, friends? Nod factors. And what's the common point? Both nodulin genes also and nod genes are also both are involved in the root nodule formation. But friends, we read that nodulin genes are exclusively present on the plant, and nod genes or nod factors are exclusively present in the bacteria. That also we finished. Later we understand how exactly the symbiotic relationship is happening. Then we produce root nodules. We also understand, friends, the root nodules contain an oxygen binding hemoprotein, which we generally call it as what, friends? Leg hemoglobin. So later, friends, we understand the real structure of the nitrogenase enzyme. Friends, please kindly don't forget the structural properties. If you quickly recall, nitrogenase enzyme contains two parts. One we call dinitrogenase reductase or dinitrogenase. Friends, always try to remember like this only. Iron sulfur redox center, molybdenum iron redox center. So friends, finally we understood that nitrogen can be converted into NH3 in how many steps we read, friends? Total 3 steps. So in this total we are using 4 pairs of electron, that is total 8 electrons, and we spent how many molecules of ATP, friends? 16 molecules of ATP. And if you all quickly recall, we can get a statement, even the asked statement in the exam. Nitrogen fixation, what do you understand by the word nitrogen fixation? Is a highly energy consuming process, which is absolutely true statement. When compared to the all other processes, we have to spend more energy in the nitrogen fixation only. So friends, this is what we studied in the last class. Now let's continue. Okay, listen to me for the period of 15 minutes. I will solve, will teach entire concept. Later we'll solve the doubts. Focus, friends. Okay, now look into the diagram. What we have studied, friends. So far we studied the first process. That is what, friends? Fixing of the nitrogen. That is conversion of NH3 or NH4. That is what we studied now. So friends, see, I hope all of you remembered. What is the ultimate goal of the nitrogen cycle? Ultimate goal of the nitrogen cycle is converting the atmospheric nitrogen into most simple forms. What are the most simple forms? NO2 and NO3. What happens when I convert the nitrogen into NO2 and NO3? So I can assimilate, I can incorporate, I can use into amino acids and proteins. Now what we're gonna do, friends, we are going to convert this particular NH3 or NH4 into NO2, and NO2 to, friends, NO3. Everyone pay attention. Now please kindly don't get confusion at all. Conversion of NH3 or NH4 to NO2 also we can call it as nitrification only. And my friends, conversion of NO2 to NO3, that is also we will be calling it as what, friends? Nitrification only. Don't get confused. See, friends, the entire process we call it as nitrification only. But friends, the type of enzymes involved are entirely different. Please kindly do not forget the statement at all. So friends, what is this conversion of NH3 or NH4 plus? First, it is going to be reduced to NO2. Then later, friends, NO2 is going to reduce to what, my friends? NO3. This is what we read. And what is the unique points all of you has to remember? The type of bacteria involved we have to remember. So friends, please kindly don't get confused with the names of the bacteria which are involved. Okay, so always remember, let's look into the bacteria here. The first bacteria, please remember, friends, in a given 4-mark question, in terms of matching we will get it. Nitrosomonas. Then friends, Nitrosococcus, don't confuse. Nitrosomonas and Nitrosococcus are the two major bacteria. Will convert the NH3 or NH4 to NO2. Now friends, NO2 basically going to converted into what, friends? NO3 by a bacteria called as, friends, Nitrobacter. And at the same time, friends, we can also call it as the Nitrococcus, right? Nitrobacter, Nitrococcus. Friends, don't confuse. Here, it is a Nitrosococcus. It is the Nitrococcus. There's a great difference between these two. And I want you to remember the direct examples of the bacteria which are involved in the production of NO2, which are involved in the production of NO3. This is a small step, nitrification. Now friends, the biggest step among all, is the assimilation. So friends, everyone pay attention from this particular slide. Friends, finally we converted N2 to NH3, NH4, NH3, NH4 to NO2, and finally to the NO3. We have done all these to make the nitrogen in a simple way. So finally what happens? Plant is going to convert all these simple forms into amino acids and proteins for its growth and development. Now the question is like this. What is the question? Suppose example, if we don't utilize the nitrogen. Listen my statements carefully. If we don't utilize the NO2, if we don't utilize the NO3, if we don't utilize the NH3, if we don't utilize the NH4, what will happen? They will be stored. Friends, that particular stored forms will become extremely toxic. So what's the statement you have to remember? Whatever the NH3, NH4, NO2, NO3 are there, friends, we should not store them under any circumstances. We have to make use of them. That is what exactly, my friends, we will be calling it as the assimilation. So let's try to understand nitrate assimilation. See, the point saying here, what happens? Everyone pay attention, friends. What happens if we store the nitrate? What happens if we store the ammonium? All of you just listen to me carefully. If I store the nitrate and if I store the ammonium, both can be toxic. There is no doubt about it. But let's understand how they will be toxic. So friends, nitrates, stored nitrates, if I don't use the nitrates and if I don't convert the nitrates, what is happening, friends? Nitrates cause a condition. What do you call it, that particular condition, friends? Methemoglobinemia. What is this particular methemoglobinemia? It is a disease in which liver reduce the nitrate to the nitrite. Friends, you know, right? When compared to the NO3, NO2 is more toxic, highly reactive. Now, what this particular NO2 will do, it will go and combine with the hemoglobin, and it will never ever allow the hemoglobin unable to bind the oxygen. Without oxygen transport, how does this particular nitrogen fixation also occur? It never ever occurs. And most important, friends, humans and other animals, it may also convert nitrate into nitrosamines. Friends, you know, right? Nitrosamines, here I have written a word, potent carcinogen. Friends, what exactly the word potent carcinogen explains us? Everyone pay attention. Potent carcinogen means at lower doses also it is having great carcinogenic activity. Carcinogenic activity means it will cause the cancer activity. Potent means lower doses. You know, no friends, whenever we are having a severe cold and allergy, I hope all of you know it, we will take the levocetirizine. How much tablet of levocetirizine we will take, friends? We will take hardly 2 mg. That's all, very low dose. In worst condition, we can increase the dose up to 5 mg. So, but you only try to imagine how much paracetamol we will take, Dolo 650 for a headache. 650 mg we will take. Dolo 650. So you have to understand certain drugs, they will show their action even at a very low doses also. Same way, friends, this particular nitrate converted into nitrosamines, which are extremely carcinogen even at the lower doses also. Now what should I do? I should not store the nitrate. Instead of storing the nitrate, what should I do, friends? I have to convert the nitrate into amino acid. Simple point. Everyone pay attention. Even I should not store ammonium also. What happens if I store the ammonium? Friends, if I store the ammonium, it will be also toxic. What happens if I store the ammonium? Friends, everyone remember, listen my statements carefully. Ammonium can destroy entire photosynthetic machinery. What do you mean by photosynthetic machinery? Light harvesting complexes, photosystem, electron acceptors, ATP synthase enzyme, all can be destroyed. Even, friends, ammonium can destroy entire electron transport chain machinery in the mitochondrial membrane also. So all of you listen, if ammonium can be stored, it can destroy entire photosynthetic machinery, it can destroy entire electron transport machinery also. So my friends, it is not at all idealized that ammonia can be stored. Now you only try to understand how ammonia is going to destroy everything. Here I have said that ammonia is going to destruct entire photosynthetic machinery. But how? Friends, all of you look into the diagram very carefully. At a high pH, ammonium combines with OH minus. See, friends, you know, right, ammonium cannot cross the membrane. If it is unable to cross the membrane, how it can destroy the entire machinery? It cannot destroy, right? So friends, how does this particular ammonium can cross the membrane? At a stroma, matrix or the cytoplasm, the pH is high, where NH4 plus combined with OH minus at the higher pH. By the loss of water, it can converted into NH3. Now the question is very clear. Now you can ask me, sir, NH4 plus is not crossing the membrane. How can you say that NH3 crossed the membrane? My friends, I can conclude the statement very clearly that NH3 can cross the membrane. How? So the question asked more than 3 times as the statements in our exam exclusively in the 4 marker. NH3 can easily cross the membrane by the aquaporins. Please kindly don't forget the statement at all. So friends, aquaporins are not only transporting the water molecule, aquaporins can also transport uncharged molecules like ammonia, urea, uric acid, glycerol, carbon dioxide. Friends, every one of you listen. Aquaporins not only transporting the water, but it can also transport NH3, it can also transport urea, uric acid, glycerol, all can be transported through the aquaporin membrane. So my friends, you can only understand NH3 finally entered inside. Now all of you understand. Inside, what's the pH, friends? Inside the pH is extremely low pH. Now what should happen, friends? This particular NH3 combines with the H plus and undoubtedly, my friends, it is going to be converted into NH4 plus. Finally, it converted into NH4 plus, then ultimately it destroy the entire photosynthetic machinery and entire electron transport chain machinery also. So what is the statement that I want to conclude here? The statement I want to conclude here is that the nitrates and ammonium, if they stored, they will become extremely toxic. So that's why plants or animals should not store these two. Instead of storing, we have to convert them into a utilizable form. What is that particular utilizable form? We can call it as that is only amino acid. Friends, from now onwards, we have to remember lot of enzyme names. Okay, and always remember, friends, try to compare with the previous enzyme. When you try to compare with the previous enzymes, it will be extremely easy to remember for the longer time. Pay attention. Nitrate assimilation. See, what you have to understand by the word nitrate assimilation, where we are converting NO3 to NO2. Later, NO2 we will convert it into NH4 plus. Later, finally we will convert it into amino acid. These are all the various steps in the assimilation. What is assimilation? I said it to all of you. Assimilation is what? Utilizing, incorporating in the body. Okay, pay attention everyone. Nitrate assimilation, incorporation of the nitrates into organic compounds like amino acid and protein. Friends, there is a lot of information to be remembered. Be patient. See here, what I am reducing to what? I am reducing, friends, listen to me carefully. I am reducing NO3 to NO2. So what is the statement you have to understand? I am reducing the nitrate to the nitrite. So my friends, the enzyme involved here is the nitrate reductase. You don't need to remember the equations at all, don't worry. What, friends? Nitrate reductase is the enzyme which is converting NO3 to NO2. And most important point you have to remember, where does this particular reaction is taking place actually? Friends, this particular reaction is taking place in the cytosol. What is the reason that this reaction has to occur only in the cytosol? The reason, friends, the nitrate reductase enzyme is present in the cytosol only. Kindly do not forget the statement at all. Now let's talk about the structure of the nitrate reductase enzyme. All of you pay attention. Now, in order to remember the structure of nitrate reductase enzyme, you try to compare with the nitrogenase enzyme. When you compare with the one enzyme with another enzyme, you can remember. Always the best way of remembering biology is comparing with one with another. By that you can easily remember. We already remember, friends, nitrogenase enzyme contain what? Iron sulfur redox center and molybdenum iron redox center. Simple point it is. Now friends, all of you pay attention. If you look into the structure of a nitrate reductase enzyme, it contains FAD. Anyway, you know to remember the structure. FAD, heme, and molybdenum associated with an organic compound which we call, friends, pterin. Don't forget, I will ask in the next class also. Friends, more information you remember, there are more chances are there that we can answer the question very correctly. Strongly remember, friends, in the competitive exams, the real wealth is the information. The more right information you remember, the more wealth we are having. The more wealth we are having in the form of information, more chances are there we can answer the question very correctly. So always try to remember. I strongly believe during the preparation, it's not at all important how much time you read. It's not important. It's only important how much you remember. Some students work really hard, extremely. They'll work super hard. But what's the use if you don't remember? So always remember, it's not important how much time you read. It's only important how much you remember. Okay, look into this. This nitrate reductase contain FAD, heme. And friends, molybdenum associated with an organic compound called as what, friends? Pterin. Very simple point it is. Now friends, listen to me very carefully. Look into this picture. Friends, once again, please try to compare. Who is the electron donor for the nitrogenase enzyme? I hope all of you remember. The electron donor for the nitrogenase enzyme is the ferredoxin. Now this is the structure of the nitrate reductase enzyme. My friends, who is the electron donor for the nitrate reductase enzyme? The electron donor is the NADH. Very simple. What the NADH is doing here, everyone look into the diagram. NADH send the electrons to FAD. FAD send electrons to the heme. Heme will send the electrons to the molybdenum associated with an organic compound called pterin. Then finally what is happening, my friends? NO3 finally get reduced to what, friends? NO2. Very, very, very simple. So friends, listen, where this particular nitrate reductase is present? In the cytosol. So friends, the production of NO2. Where does this production of NO2 occurred? In the cytosol. But you only tell me, do you think we can store the NO2? No, not at all. Friends, just now I said to you, NO3 only toxic. But when compared to the NO3, NO2, that is the nitrite, it is little more toxic. Very highly reactive. That's why plants should not store them. So they have to convert this particular nitrite to what, my friends? Ammonium. Please don't forget the statement. Nitrite is a highly reactive and toxic ion. Never forget that, friends. All of you listen to me. Now what I am doing? I am converting NO2 to NH4. I hope all of you remember. Friends, where this particular NO2 is produced in the cytosol. Now friends, this particular NO2 will transport from the cytosol. Where it is coming, friends? From the cytosol, it is coming into the chloroplast of leaves and it is coming to the plastids in the root. Now you can ask me, but why it is coming? The reason it is coming to the leaves and plastid, the nitrite reductase enzyme, it is present in the chloroplast of leaf. It is present in the plastids in the root. That's why they have to come. Now friends, once again, please listen. You have to remember what? The electron acceptor. Electron donor. Electron donor, we already read, right? For the nitrogenase enzyme, what we read? Ferredoxin. Just now we read the electron donor for the nitrate reductase is what we read? NADH. My friends, please listen to me carefully. Once again, the electron donor for the nitrite reductase is what, friends? Electron donor for the nitrate reductase will be the same FD only, ferredoxin only. Don't forget and always remember. So NO2 can be converted into NH4 plus. You don't need to remember the equations, don't worry at all. Jayashree, look into this dear. We are converting ammonium also amino acid. Still the process is not completed, right dear? Look into it, look into it. You will understand. So NO2 is basically converted into NH4 plus. Okay. Now what is the structure of nitrite reductase? Once again, every one of you understand. Try to compare once again. What does nitrogenase enzyme made up of? Iron sulfur redox center, molybdenum iron redox center. What does the nitrate reductase is made up of? FAD heme and molybdenum associated with an organic compound called pterin. Friends, when you look into the structure of a nitrite reductase, if you look into the structure of nitrate reductase, it's a simplified structure. It just contains an iron sulfur cluster, then friends, followed by the specialized heme. That's all it contains. What does it contain? An iron sulfur cluster and specialized heme. This is the simplified structure. And friends, who is the electron donor? Once again, undoubtedly my friends, the electron donor will be the ferredoxin. So ferredoxin will donate electron to iron sulfur cluster to heme, heme to NO2, NO2 is getting reduced to NH4 plus. Okay, now look into Jayashree. Of course, whether it is the NO3 or NO2, finally we converted into what? Ammonium. But what is the use if I convert into ammonium? Ammonium is also a toxic form only, right? So I cannot store the ammonium also. What is my ultimate goal? Whether it is a NO3, whether it is the NO2, whether it is the ammonia, whether it is the ammonium, my ultimate goal is I have to convert them into amino acids. Now finally I am doing the process of ammonium assimilation, incorporating the ammonium into amino acid, converting the ammonium into amino acids. So in this particular nitrate assimilation we produced ammonium. See, all of you understand, friends. Listen the statements carefully. Plant cells rapidly convert the ammonium generated from the nitrate assimilation or photorespiration into what, my friends? Into amino acids to avoid what? Toxicity. Friends, during the process of photorespiration, I don't know whether you recall or not, especially in the organelle mitochondria. In the mitochondria, what is happening? I hope all of you remember, my friends. When the amino acid glycine can be converted into another amino acid serine, during this particular time, my friends, NH4 will be released actually. So all of you listen, whatever the NH4 that is produced here and whatever the NH4 just now we produced by the conversion of nitrate, this entire ammonium we are not going to store. We are going to convert into amino acids. Good morning all. Please continue. Now everyone pay utmost attention towards this. The primary pathway for this particular conversion involves two major enzymes. Everyone, one we called glutamine synthetase enzyme, and my friends, another one we called as glutamate synthase enzyme. Friends, from here please kindly understand every single point without confusion. I'll make it very simple. Listen. Glutamine synthetase. Friends, what is the difference between an enzyme synthetase and an enzyme synthase? Always keep it in your mind, friends. When you read synthetase, keep it in your mind, it always uses ATP. There is no doubt about it. When we use the word synthase, always remember it won't use the direct sources of ATP. So maybe it will use the NADH, or if it may use FADH2. But the point is, my friends, synthases never use the direct sources. Synthetase always uses the direct sources. That's a simple statement. Now friends, don't confuse once again. Glutamine synthetase. What the glutamine synthetase enzyme will do? It will synthesize the glutamine. Glutamate synthase. What it will do? It will synthesize the glutamate. Let's see how. Is there any necessity to remember the structure? Not at all. You never gonna get in the exam. Okay, but try to understand the sequential step. Everyone pay attention, friends. Glutamate, what we have right now? We have NH4 plus with us. We are converting this particular NH4 into amino acid. All of you pay attention, look into the equation. Ammonium, everyone friends, ammonium combined with the glutamate. And we are converting into what, friends? Glutamine. So now the question is very clear. How this particular glutamine can be synthesized? By an enzyme called as a glutamine synthetase enzyme. Since I told you, friends, it is the synthetase enzyme, undoubtedly, friends, it is going to use the energy directly. Now you see now, glutamate having an extra amine group, right? That extra amine group is donated by the ammonium. Very simple point, and you don't need to remember that. What I want you to remember is that how does this glutamate is converted into glutamine? What is the enzyme catalyzed that particular reaction? And friends, the enzyme involved is the glutamine. Please remember for the exam, if this particular glutamine synthetase wants to be active, it requires major cofactors. What are the major cofactors required? Magnesium, manganese or cobalt. These are all basically acting as the cofactors. I want you to remember this. So friends, now what is the next enzyme? This is the first enzyme we studied, glutamine synthetase. My friends, what is the next enzyme? Glutamate synthase, or what, my friends? GOGAT. You people no need to remember the full form of the GOGAT, it's not required. Just remember, my friends, GOGAT, that's all. Now just listen to me. Right now what we have done, just now what we done in the previous step. Listen everyone, listen. In the previous step, I have converted the I have added ammonium to the glutamate and I converted into glutamine. That is what I have done in the previous step by an enzyme glutamine synthetase. Now I produced the glutamine, right? Now this glutamine, what I have done, I have combined with, friends, 2-oxoglutarate. Why I have combined the glutamine into 2-oxoglutarate? In order to produce 2 glutamates. My friends, so what is the major enzyme involved here? Obviously GOGAT. Friends, I have told you very clearly, glutamine synthetase using the energy from ATP. You know the speciality of this particular GOGAT enzyme. This GOGAT enzyme use the energy from two sources. Either it can use the energy directly from NADH, friends, or it can use the energy from the ferredoxin also. I am summarizing the equation for all of you. See, in this reaction you can observe one great point. What is that great point? By using one glutamate, friends, everyone pay attention. By using one glutamate, we can produce 2 glutamates. Do not forget the statement. Now what's happening here, friends? Ammonium combined with the glutamate, we produced the glutamine. By combining the glutamine with 2-oxoglutarate, we are producing 2 glutamates. Ultimate goal is what? Production of the amino acid. Now my friends, ultimately we have converted or we have assimilated the ammonium into amino acids. As I said to you, the GOGAT will do or will use the electron from 2 sources. Just you people remember, GOGAT accept electrons from NADH also, friends, and even GOGAT accept the electrons from the ferredoxin also. There's no difference. Equation is entirely same, friends. Only difference is the electrons. So glutamine 2-oxoglutarate, electrons given by the NADH. Reaction is same. Same glutamine, same 2-oxoglutarate, electrons given by the ferredoxin. There will be no change at all. So I want you to just remember, GOGAT will exhibit it in two forms. It can accept NADH also. It accepts the electrons FD also. So friends, finally what we have done, we converted NO3 to NO2, NO2 to NH4 plus, NH4 plus. Finally we have converted into amino acids. We have fulfilled the goal of the assimilation. We did not store anything, right? We did not store anything. Instead of storing, we converted them into a utilizable forms. What are the particular utilizable forms? Nothing but amino acid form only. Finally, we produced amino acids. There's no doubt about it. Now, what is the reactions we called, friends, transamination reaction? Why this particular transamination reactions has to take place? In this reactions, friends, we are converting one amino acid into the another amino acid. But how? Just by the transfer of nitrogen. Because friends, we produced only one or two. We produced glutamine and glutamate, right? But glutamine and glutamate are the not only amino acid. There are many other amino acids also there. So we are converting one amino acid to the another amino acid by a simplified transamination reaction. Do not forget it. Once assimilated into glutamine and glutamate, nitrogen is incorporated into other amino acids via transamination reactions. Now let's take a wonderful example. The enzyme involved here, my friends, you have to remember this. The example given here is aspartate amino transferase enzyme. What is this particular aspartate amino transferase enzyme will do? It see here, glutamate combines with the oxaloacetate. Oh, there is an alarm sound is coming. 1 second. Sorry. When the glutamate combine with the oxaloacetate, immediately what is going to happen, friends? See here what is the enzyme involved? The enzyme involved is the aspartate amino transferase enzyme. Now this aspartate amino transferase enzyme function is what, friends? It will just transfer this amino group to the aspartate. See, now glutamate converted into aspartate, and what is this oxaloacetate converted into 2-oxoglutarate. You no need to remember which amino acid is going to be converted into which amino acid. Also, you no need to remember. What you have to remember is that one amino acid can be converted into another amino acid by the process of transamination. That's the only point I want you people to remember. See here by using aspartate amino transferase enzyme, I have synthesized aspartate. Now let's take another enzyme, asparagine synthetase enzyme. By using asparagine synthetase enzyme, friends, we are going to produce the asparagine. Same reaction. Glutamine, everyone friends focus. Glutamine combined with the aspartate, giving rise to what? Asparagine. See, glutamine having one amino group. See, asparagine having the two amine groups. And glutamine, upon donating this particular amine group, it is converted into glutamate. Glutamine donate this amine group to the aspartate. Upon accepting the amine group, aspartate converted into asparagine. Very simple. Glutamine donate the amine group, converted into glutamate. Upon accepting the amine group, aspartate converted into asparagine. No need to remember. What I want you to remember, asparagine synthetase is the enzyme which is only involved in the synthesis of asparagine. So friends, this is the biggest step of the nitrogen cycle. That is what, friends? Assimilation. One of the most important step I can say. So friends, what are the steps that we have studied so far? We converted atmospheric nitrogen to NH3NH4. NH3NH4 we converted to NO2 and NO3. Ultimately, we have assimilated NO2 and NO3 in the plant body. Now, what's the major step? Now you listen. Who will utilize it right now? Plant utilized the amino acids. So plant utilize this particular NH3, NH4, NO2, NO3. They produce amino acid. They produce proteins. Listen every one of you friends. Upon eating the plants, we animals also will get the proteins and amino acid. There is no doubt about it. Now all of you pay attention. What happens when the plant died? When the animal died, what is happening friends? When the plant died and animal died, what will happen friends? The dead organic matter of a plant and animal decomposed by whom friends? They are decomposed by the decomposers. Friends, what the decomposers will do? All of you know it. Decomposers will convert the organic to inorganic. What are organics? Proteins, amino acids, nucleic acid. These are only organic, right? These all organic can be converted into what? Inorganic. Carbon, hydrogen, oxygen, nitrogen. That's all. Now what the decomposers like bacteria is doing? They will take, see, I am a bacteria. Listen, I am a bacteria. I am a decomposer. What I will do? I will convert the dead organic matter into inorganic like carbon, hydrogen, oxygen, nitrogen, and I will use that particular nitrogen. Why will I use that particular nitrogen? I will use that particular nitrogen to produce proteins and amino acids for me. And okay, that's fine. But what I will do with the excess nitrogen? Friends, that excess nitrogen I am going to convert in the form of ammonium. So listen my statements everyone. Fixing of the excessive nitrogen into the ammonium, my friends, we will be calling this particular process as ammonification. Do not forget the statement. What do you call this particular process? Ammonification. Everyone look into this. Death of plant and animals. Once the death occurred, all the organic compounds like protein, amino acids, nucleic acid, nucleotides are released into the soil. What happens now? The decomposers break down this all organic compounds into inorganic. Now they will use that nitrogen and they will fix that excess nitrogen in the form of ammonia. This is only we call, my friends, ammonification. So friends, now what are all developed friends? Now you only try to understand plant benefited, animal benefited, decomposers also get benefited. Friends, from where we have taken the nitrogen? We have taken the nitrogen from the atmosphere actually. Is it? So friends, we have to send this particular atmospheric nitrogen, we have to send back the nitrogen to the atmosphere. So friends, what we are doing now? Conversion or transfer of NO3 back to the atmospheric nitrogen will complete the nitrogen cycle. Once again understand friends, conversion of NO3 back to the atmospheric nitrogen will complete this particular nitrogen cycle. This is what do you call, my friends? Denitrification. So who's gonna do this? Just remember, denitrification means conversion of nitrates back to N2 by releasing NO2. This is called denitrification. It can be mediated by the denitrifying bacteria. So just remember only 2 points. The examples are Pseudomonas clostridium. Just remember that. So that ends the nitrogen metabolism chapter. Now you can ask me any kind of a doubts. Tarun, once again you are missing the logic. Tarun, you look into this. I understand your doubt. Once excess nitrogen can be converted into NH4 plus, Tarun, once again it is converting into NO2 and NO3, and once again it is getting assimilated, right? It is not getting stored. You're getting my point. Once it converted into NH4, once again it will be converting into a simple forms. It's not gonna store it. Okay, friends, is that clear for everyone? Please let me know. I will help if anyone having any kind of a doubts. Is it clear? Come on, be active. Clear for everyone? Okay. I hope it's clear for others. Okay. So friends, right now what we have completed is we have completed entire photosynthesis concept, and friends, we have completed the nitrogen metabolism concept also. I'm requesting all of you friends, please keep on revising. Keep on revising. Yeah, Reens, we studied the same thing in 10th standard, na? Cytoplasm and cytosol. Cytosol means without organelles. That is called cytosol. Cytoplasm means with organelles. Ritika, nitrocystis can be used in the NMOX process, Ritika. That NMOX we will discuss in the ecology chapter. It is used in the NMOX. Is that clear? Okay. Reens, cytoplasm means with the organelles. Cytosol means it's only fluid without organelles. Okay. So friends, I am requesting all of you, please revise this without fail. Okay. So once the chapter is completed, immediately I'll keep on solving all the questions. Okay. So friends, finally we have completed 2 subunits. Now friends, we are going to start the third subunit. What is that, friends? Plant hormones. I'll finish the syllabus very faster this time, but in a very effective way. Okay. Now, my only sincere request to all of you, just be regular. See, I strongly believe if we want to be succeeded in the competitive exam, friends, the excellent key to our success is the consistency. Whomever are really consistent, undoubtedly they will become successful. You have to believe success really becomes our slave if we are really consistent in our work. Okay, so I am requesting everyone, please put at least 4 to 5 hours of dedicated preparation every single day without missing. See, in order to get something, you have to sacrifice something. Okay, here you are not sacrificing anything for the bad purpose. You are sacrificing something for a wonderful future. Okay, so I'm requesting every one of you, make use of your utmost time and spend quality time on your preparation, devoted time. Okay. Okay, now let's talk about, friends, this particular plant hormones. You all commonly know that plant hormones is the topic which is extremely important topic, friends, even for the part B also, friends, even for the part C also. Very important, friends. Okay, and for the plant hormones, the PPT will helpful to you 90%. Okay, along with the PPT, I encourage all of you to just read once Pathfinder, just once. Read the Pathfinder once and just revise that. That's all. Now, friends, you know, before 2015 December in the question paper, they never ever used to ask any kind of a question from the signaling at all. They never used to ask. But friends, after that, every single time they are asking the questions from the signaling also. You know, plant hormones, secondary metabolites, stress physiology, sensory photobiology, transport in plants. These five topics are informative topics, which means a lot of information to be remembered. At the same time, lot of information to be understood also. So let's talk about all the hormones. Every single point we have to read in this particular hormones. Okay, so friends, let's talk about the hormones. How can we define the word hormones? How can I define the word hormones? Friends, listen everyone. Hormones are the chemical messenger. I'll explain from basic to the advanced level. Just pay attention. Chemical messenger. What do you mean by the word chemical messengers? Which can carry the message, which can carry the information from one area to the another area. All of you pay attention. Second point, friends, plant hormones are non-nutritious in nature. Of course, true, right? Hormones do not have any nutritional value. And the unique statement every one of you has to remember for the exam point of view, friends, plant hormones produced in a very low concentration. My friends, what do you mean by that? Which means low the concentration, high the activity. Always remember, low the concentration, high the activity. High the concentration, low the activity. Every time remember that particular statement. If a hormone is present in the plant body or even in the animal also, you take any organism, hormone, if it is in the low concentration, show the better activity. If the hormone is present in the high concentration, show very low activity. And one of the most significant point I want every one of you to remember for the exam is site of production and site of action is entirely different. Friends, what is the difference here? What is the meaning of this point? Site of production and site of action is different, which means everyone understand. Hormone produced at one area. Hormone produced at one area, but friends, hormone is going to show their action on the another area. Let me tell you, you take human body only. In case of a hypothalamus, just I am telling you an example. Hypothalamus can synthesize a GnRH hormone, correct, right? Gonadotropin releasing hormone. And we also know that the GnRH further give rise to FSH and LH. FSH and LH further involved in the production of the estrogen and progesterone. My friends, the estrogen and progesterone production started at the hypothalamus. But do you think estrogen and progesterone are showing their action in the hypothalamus? They will act on the ovaries during the menstrual cycle. So what's the point you have to understand? Site of production, site of action is entirely different. Not only in the animal, even in the plant body also. Some hormones synthesized in the shoot, they are going to show their action on the root. Some hormones synthesized in the root, they are going to show their action on the leaf. So that's why site of production, site of action is entirely different. Do not forget that. Now, friends, what are the various hormones that we have to read? Okay, based on what criteria we categorize the hormones? First of all of you pay attention, friends. We categorize the hormones into different types based on exclusively whether they are promoting the growth or they are inhibiting the growth. Suppose example, if they promote the growth, we will be calling them as the growth promoters. Friends, if they inhibiting the growth, we will be calling them as the growth inhibitors. I want you people to remember every possible example, friends. Growth promoters are what, friends? Auxin is the growth promoter, no doubt about it. Cytokinin is also a growth promoter, no doubt about it. And friends, gibberellic acid is also a a growth promoter. There is a no doubt about it. And friends, what is the statement you have to remember? What about the growth inhibitor, my friends? All of you listen. The growth inhibitor, one of the major growth inhibitor in the plant body is the abscisic acid. No doubt about it. And friends, what about the another fifth hormone, ethylene? Friends, you know, right? Ethylene is a gaseous hormone present in the plant body. I want all of you remember, friends, ethylene can act as a growth promoter also. Ethylene can act as a growth inhibitor also, depending on the condition, depending on the situation. So, listen everyone. Auxin, cytokinin, gibberellic acids are the promoters which can enhance or promote the growth. Abscisic acid is the one which inhibit the growth. Ethylene is the one which is acting as a both growth promoter also, friends, growth inhibitor also. Do not forget the statement, please. Now friends, let's talk about one by one hormone elaboratively. Before that, friends, it is a very important to remember the derivatives. Okay, so friends, everyone pay attention. What are the derivatives? You can ask me, sir, is there any necessity to remember the derivatives? Of course, my friends, it's a highly necessity to remember the derivatives, which we can expect directly in the 4-marker question itself. So let's try to understand which hormone is derived from what. Let's consider auxin. Auxin, what do you call auxin? We can call auxin is the indole derivatives. You know, no friends, we will read it. Indole acetic acid, indole butyric acid we called. So from where they are derived? From indole derivatives. Friends, everyone listen. Gibberellic acid. It is exclusively derived from what, my friends? Terpenes. What are terpenes? We are going to read in the secondary metabolites elaboratively. You know, right? Even from the terpenes also, we got 4-marker question in this 2022 February question paper. Gibberellic acid is derived from what, friends? Terpenes. Now next one, cytokinin. Friends, please remember, cytokinin is basically derived from what, friends? Purines. They are derived from what, friends? Purines. Example of purine, especially my friends, cytokinin is derived from adenine exclusively. Then friends, what is the another hormone is there now? Abscisic acid. Friends, please don't forget. Abscisic acid is exclusively derived from the carotenoids. Please don't forget this. Then friends, finally, the one and only hormone in the plant body which is gaseous in nature. What do you call it that, friends? Ethylene. Ethylene is the only hormone which is a gaseous in nature. I don't know what you do, friends. It's very important. You have to remember the derivatives. Here you no need to use any kind of a intelligence. Here you have to just remember it for the long time. That's all. So auxin derived from indole, gibberellic acid from terpenes, cytokinin from purines, abscisic acid from carotenoid, ethylene is from the gas, which is a gaseous hormone. Simple point. Now friends, let's start with one by one hormone elaboratively. So let's start with the first hormone, auxin. So friends, I will explain everything briefly, friends. Just pay attention. The only thing I want you to focus. Before starting the auxin, let's try to understand briefly about discovery of the auxin. It is very important you people have to know how the auxin got discovered. See friends, just listen to me carefully. In the year 1880, two intelligent scientists, I will explain point by point, pay attention. In the year 1880, the leading scientist, Charles Darwin, and his son, Francis Darwin. Friends, what they have done, they started doing the experiments on the coleoptile. Friends, on where they are doing the experiments? Coleoptile. Now you can ask me, sir, what is coleoptile? How can you define the word coleoptile? Suppose friends, this is the shoot. This is the shoot. You know, the protective cover, everyone listen to me carefully. The protective cover or the protective sheath, the protective cover or the protective sheath of the shoot we called coleoptile. Very simple point. So friends, whenever they are doing the experiment to the coleoptile, you know what they have done? They have exposed the coleoptile to the unilateral sunlight. Friends, what do you mean by the word unilateral sunlight? The movement of sunlight in only one direction. Very simple. They have taken the coleoptile, they have exposed the coleoptile to the unilateral sunlight. My friends, when he exposed the coleoptile to the unilateral sunlight, all of you pay attention. Coleoptile starts bending like this. Coleoptile start bending like this. Now, what Darwin and his son concluded finally? Darwin and his son concluded one important point. What is that point? Both said there is a chemical present. Listen, every one of you, my friends. There is a chemical present in the tip of the coleoptile. Listen to me. There is a chemical present in the tip of the coleoptile. Upon perceiving the light, that chemical start moving from lighter region to the darker region, and it is causing the coleoptile to bend. Once again, listen. Darwin and his son concluded that there is a chemical present in the tip. That chemical, upon exposing to the sunlight, moving from lighter region to the darker region and causing the coleoptile to bend. Unfortunately, friends, Darwin and his son, they did not get any credit. Why they did not get any credit? Because they did not explain what chemical it is, from where it is moving, okay, and how it is moving, how it is elongating the coleoptile, how it is bending the coleoptile. They did not explain anything. That's why these two scientists did not get any kind of a credit at all. Later, very intelligent scientist named as Francis Went, or you can call it F. Went. He is the one finally proved everything. Just listen to me carefully, just understand. Francis Went is the scientist who proved or who isolated the chemical auxin. Everyone listen, same thing he also done, friends. He has taken what, friends? He has taken the coleoptile. Okay, he has taken the coleoptile. My friends, every one of you listen to me carefully. From where he has taken the coleoptile? That is the point. He has taken the coleoptile, friends, from the Avena grass seedlings. From where he has taken? Avena grass. What is Avena grass? Common oat plant. Correct, right? We will eat oats. That oat only we can call the Avena grass. Friends, every one of you pay attention. It's such a beautiful experiment it is. He has taken the coleoptile. He has exposed the coleoptile to the unilateral sunlight. Friends, whenever he exposed the coleoptile to the unilateral sunlight, what he observed, my friends, of course, the coleoptile starts bending. There's no doubt about it. Now, once again, what he has done, my friends, he has taken the coleoptile. This time, you know, my friends, what he has done? He cut the tip. What he has done? He just cut that particular tip. What do you mean by the word cut the tip? See, this is how the coleoptile looks like, right? He cut the upper tip. So whatever the tip he cut, he kept in the agar solution. Where he kept? In the agar solution. Just for temporarily he kept in the agar, that's all. Whichever you kept in the agar, anyway agar diffuse it. Just he kept in the agar solution. Now without cap, how does it look like? It looks like this. My friends, everyone pay attention. Without cap, what do you call it as? Without cap, you will be calling it as the decapitated part. What do you mean with the word decapitated? Without cap. My friends, now you know what he has done. Without cap, he exposed to the sunlight. Without cap, he has exposure to the unilateral sunlight. Do you think it starts bending? Not at all. It's not at all bending. Now, after some time, what the vent does, once again, whatever this particular cap he kept in the agar solution, my friends, he put it back on this particular decapitated part and exposure to the unilateral sunlight. My friends, then this particular coleoptile starts bending. Now friends, what is the conclusion that I can come now? The conclusion that I can give you is that yes, there is a chemical only present at the tip of the coleoptile only, not anywhere else. That chemical moving from light to the dark region like this exactly when they expose it to the sunlight. You know what it is doing? Whenever it is coming from light to dark region, I will show you in the next slide. It will cause the cell to get elongated. What it will cause? It will cause the cell to get elongated. Because of that elongation, coleoptile start bending. Once again, quickly look into this. First, vent has taken the coleoptile. He has exposed the coleoptile to unilateral sunlight. Then coleoptile starts bending. When the coleoptile starts bending, what happened, friends? It exposed this particular, he cut the tip of the coleoptile. Whatever the tip he cut, he kept in the agar solution. Without a tip or without cap, we can call decapitated part. That decapitated part, he exposed it to the unilateral sunlight. My friends, then it starts showing bending. So what he concluded, there is a chemical present in the tip. He is the one, cis vent is the one who isolated that chemical, given the name auxin, which means he has given a definition. Auxin means to grow. Friends, basically he has isolated auxin from where? From the Avena grass seedling. Correct na? So that's why he has given the name for this particular test as Avena coleoptile curvature test. Avena coleoptile curvature test is the confirmatory test for the presence of auxin. Always remember it. He is the first scientist who isolated the auxin. After him, my friends, there is another scientist named as Kogel, K-O-G-L. Kogel is the scientist who isolated the auxin from the human urine. My friends, please listen my statements. You should not think that auxin have a key role in our body. That is absolutely wrong statement. Actually, at a very, very low concentration only, friends, auxin was identified in the human urine. But till today, we don't know exactly what is the role of the auxin in the human urine. Till today, we don't know actually. Remember, friends, don't forget that. This is what a simplified experiment performed by the cis vent to prove the existence of auxin. Now let's look into it. What are the types of auxin? Okay, there are many types are there. Natural auxin, synthetic auxin, free auxin, bound auxin. There is another type also there, my friends. We will be calling it as the anti-auxins. Friends, now let's talk one by one briefly. First one, natural auxin. How can I define the word natural auxin? The name itself indicating, right? Whichever synthesized naturally in the plant body. That's what we call natural auxin. What are the best examples, friends? One is the IAA, indole acetic acid, and my friends, indole butyric acid. Simple point it is. Next one, friends, synthetic auxin. Name itself indicating which are synthesized or artificial or man-made. Best example, you have to remember example, NPA, naphthyl thalamic acid. Friends, please remember the examples. 2,4 dichloro, 2,4 dichlorophenoxyacetic acid, 2, 4, 5 trichlorophenoxyacetic acid. Very simple, right? 2, 4 are the carbon number where chlorofunctional groups are present. 2, 4, 5 are the carbon number where the chlorofunctional groups are present. My friends, even the question asked in the exam also, these two synthetic oxygens, friends, act like a powerful VD sites. Powerful VD sites which will kill the weeds. Now friends, don't get confused between the word free oxygen and antioxidant. All of you listen my statements very carefully, friends. If I isolate an oxygen without organic solvent, if I want to isolate an oxygen, I did not use any kind of organic solvent. Very easily I have isolated. That kind of oxygen called as free oxygen. Now you can ask me, sir, give me the example for the free oxygen. I can tell to all of you, a free oxygen, it may be natural oxygen also. A free oxygen, it may be synthetic oxygen also. But if this particular oxygen wants to be isolated, if we don't use any organic solvent, we can call it free oxygen. Then what do you mean by the word bound oxygen? If we want to isolate this particular bound oxygen, we have to use the powerful organic solvents like acetone, ethanol, carbon tetrachloride, chloroform, all this. Now if you ask me examples of the bound oxygen, what I can tell, friends, a bound oxygen may be natural oxygen also. A bound oxygen may be synthetic oxygen also. It may be anything, my friends. If we use the organic solvent for its isolation, we can call it bound oxygen. Very simple. Now friends, antioxidant, name itself indicating, right? Anti again is the oxygen. So every inhibitor which is inhibiting the oxygen, we can call it this as the antioxidant, right? Okay, I hope everyone is clear till this particular slide. We will continue remaining in the tomorrow class, that is the day after tomorrow. So tomorrow will be continuing the molecular biology transcription. Is it clear, friends? Please let me know quickly, please. I have another class. Is it clear? Okay, great. Okay. So friends, that's all for now. You can download the PPT after 12:30 or 1 p.m. You can download afternoon. Okay. Okay, friends. That's all for now. I'll meet you in the tomorrow morning class. Thank you so much. Thank you all of you. Thank you. Bye bye. Thank you. Thank you all.